Okay, so good morning, everyone. I, Abhishek Das, feel honored to welcome you all to what promises to be an enlightening and transformative journey into the world of bioinformatics. On behalf of the principal, CEO, and management of Patkar-Varde College, I, Mr. Abhishek Das, feels delighted to welcome you all to the RUSA-sponsored national webinar on bioinformatic tools for epigenetic research in neurodegenerative diseases, a multidisciplinary approach organized by the Department of Bioinformatics. I welcome our Chief Education Officer, Dr. Mala Kharkar, our Principal, Dr. Pratibha Gaikwad ma'am, Chief Coordinator of Sciences and Coordinator of Bioinformatics, Mrs. Ruchita Delvi ma'am, our sponsoring organization, RUSA, and all teaching faculties and students. We are honored to have a distinguished guest, Dr. FARHAN Zameer, with us today, who will share invaluable insights and uh, perspectives on neurodegenerative disorders. We are delighted to have our beloved principal ma'am, Dr. Pratibha Gaikwad, amongst us. I would like to introduce our respected principal, an academic and administrative leader, author, resource person, obtained her MA Economics from Wadia College, Pune, and PhD from Pune University. She has 27 years of teaching experience as a PhD research guide. 12 students have obtained PhD under her guidance, and 4 students are pursuing PhD. She has been conferred on 2 design patents by Government of India in the capacity of NAC peer team member. She has assisted 40 plus colleges throughout India. Ma'am, we are grateful to have you today with us. I would request ma'am to share a few words.
Thank you, sir. On behalf of Chikitsak Samuhas, Patkar-Varde College, it is a matter of great privilege to have amidst us a very enlightened speaker, Dr. FARHAN ZAMEER, Professor and Chief Scientific Officer from Karnataka. Sir, your enlightened speech will indeed be a matter of great privilege and addition to the knowledge of our existing very enthusiastic faculty as well as the students. Under the aegis of RUSA, Rashtriya Uchchatar Shiksha Abhiyan, this national webinar has been organized, and I, on this behalf, heartily congratulate the Department of Bioinformatics who is working under the able leadership of our management in general and specifically under the able guidance of Dr. Mala Kharkar Madam. Mrs. Ruchita Dalvi, my colleague, has put in her best efforts in order to make this national webinar happen. Sir, with these few words, this college I would like to bring to your notice last year has celebrated its diamond jubilee year, and we have number of very applicable programs running in the college which is of great importance and which are in tune with NEP 2020 expectations. Now this topic, as rightly introduced by the speaker, my colleague, I hail from the subject economics. So as a layperson, as a beneficiary of bioinformatics and AI tools, which helps to improve human life. These topics are of great importance, and research and development should be enhanced in such topics, which will enhance the longevity and the health of people in India and abroad. So, with these few words, I once again wish the conference, the national webinar, a great success, and I encourage my faculty, staff, and students to greatly benefit from this national webinar and keep on improving the quality of higher education in general and especially research which will help in enhancing human physique in specific. So I welcome you all and I wish this national webinar a great success. Thank you very much.
Thank you, ma'am.
Thank
Now,
you.
I would like to introduce our speaker for today, Dr. Farhan Zameer. Dr. Farhan Zameer is presently serving as Professor, PG, Department of Ayurveda Pharmacology, and Chief Scientific Officer, Atma Research and Development at Alva's Ayurveda Medical College, Vidyagiri, Mudbidri, Dr. Zameer has served in capacities both academic and administrative in India and abroad. He has good experience of 15 years in teaching and 18 years in research. Dr. Zameer has completed his postdoctoral studies from the School of Medicine, Department of Pathology, Microbiology, and Immunology, Center, University of South Carolina, USA. He is conferred with Sir C.V. Raman Fellowship from the University Grants Commission, New Delhi, Government of India, to pursue research in USA with a specialization in biochemistry, bacterial thiomics, epigenetics, anti-inflammation, microbiome, and neurodegenerative diseases, that is multiple sclerosis. Dr. Zameer has completed his PhD from the University of Mysore in biochemistry with ICMR, BMBF, Indo-German Israel Fellowships to study thiomics, bacterial signaling, and gene expression studies. He has a master's degree from Mangalore University with first rank, a gold medal in biochemistry, and bachelor's degree from Kuvempu University. Further, he has qualified Indian Council of Medical Research and UGC KSET exams. He has also completed Master Facilitator certification in Leadership and Human Resource Development from College for Leadership and Human Resource Development, Mangalore, and PG Diploma in Information Technology from Manipal. His area of research interest is in the field of biochemistry of thiol gene expression, protein enzyme studies, regulation of metabolic pathways with special reference to cellular oxidative stress, biocrines, and cellular signaling pathways, including bioinformatics, approaches, and epigenetics, mediated mechanisms in inflammatory fly and mouse models, and the related microbiome analysis. He is also part of various collaborative research projects with scientists of eminence in organizations of repute. He is a reviewer for several national and international publishers. Dr. Zameer is a life member of various scientific societies, SLB USA, EUPMBR Germany, Chinese Chemical Society China, SCA, SBC, and many such organizations. So currently, 4 students have completed PhD program in biochemistry under his mentorship and have published 151 plus research papers and 5 patents, Canadian and Indian patents. He is well recognized for the structurization of new proteins called claudins in Drosophila. Humans, their role in neurodegenerative disorders. He has authored 5 books and more than 30 book chapters published by national and international book house. He is applauded for his research contribution, which is evident from the various Young Scientist awards, honors, government fundings, and citations received by him. Sir, it is indeed a pleasure to have you with us today. Before we begin with the webinar, I request all the participants to switch off their cameras and speakers so to allow smooth conductance of the webinar. If any technical problem arises during the webinar, we will try to resolve them. Kindly post your questions in the chat box. It will be answered at the end of the sessions. Now, I would request Sir to address the webinar.
Thank you very much, sir. Hello, everybody. Very good morning. Hope I am very audible. And in a minute, let me try to share the screen and then we will start. I need to thank many people, before I forget. I need to thank the principal of the college who has given me an opportunity to share ideas of whatever we have, whatever we are learning from past 2 decades. Special thanks to Ruchita Delvi ma'am. Because of her correspondence and the entire process scenario, I am here for today's talk. A very special talk, thanks to my student, Grace Matthew, who actually recommended me to deliver this particular talk, and I'm happy that Grace has connected me to a right place. Looking into the entire paraphernalia and the history of the college, I'm very happy to know from the principal that this is a NAC graded college, and the entire fraternity, life sciences and non-life science people, they're coming together to learn something new, so that we nurture the young minds of the future India. With all this, I thank everybody who are directly and indirectly connected in the execution of this particular webinar. So let's dive into the today's topic. I hope that there are no technical glitches. And any point of time, if you feel that there is some problem, technical glitch in between, please stop me so that I can rectify and then move forward. Now, coming on to today's talk, we are starting exactly at around 11:45. I'll make sure that the talk would be delivered for 45 minutes and then followed by another 15 to 20 minutes of the discussion. Fine. So, coming on to today's caption of the webinar, which speaks on the bioinformatic tools for epigenetic research in neurodegenerative disorders, a multidisciplinary approach. As I have been rightly introduced, I am FARHAN ZAMEER, and for the next 1 hour, I will share my thoughts of our learning from past 20 years. I hope there's a change in slide. Please confirm if it is yes. Can anybody confirm that there's a change in the slide?
So slide is not visible.
Slide is not visible. Okay. So is it visible now? So is it visible now?
No, sir. Yes, sir.
Please check.
It is visible. It is visible.
Okay. Now I have changed my slide into slide number 2. Is it visible?
Yes, sir.
Okay, great. So from this point onwards, I'll be continuous and hope so the internet also favors us with all uninterrupted connectivity. Now, coming on to biotechnology. So I'm sure that most of the students who are attending are biotechnologists, and we are into a beautiful era of biotechnology, wherein there is an amalgamation of various disciplines, so that multiple brains, they come together so that something new could be explored. Now, as in biotechnology, we are trying to amalgamate biology with that of technologies, and this will facilitate the better understanding of biology, so that we can minimize the suffering in terms of human health. We can minimize suffering and create a holistic approach, so that people all around the globe can stay happy for their whatever tenure they are all assigned onto this particular planet Earth. So biotechnology would play a very primordial role, and it is a promising subject in terms of accelerating scientific research in life sciences. Now, I am not here to define what exactly is biotechnology. I just wanted to make you understand, the people who are attending from a non-science background, that this is a promising area which has a combination of both biology and technology, and we are trying to understand biology using the aid of technology. Now, just to have a brief on what exactly are the various kinds of applications in terms of biotechnology. Biotechnology could be used in agriculture, nutrition, health and medicine, industry, then environment, marine, and then various other, even madam was from economics background. So here comes a very important role of the law and ethics, okay, which plays a very, very crucial role in patenting and IPR issues related to biotechnology. However, I want you to drag your attention on to today's important domain of biotechnology, that is health and medicine. Let us try to understand this, the application of health and medicine, and how best we can dwell deeper into this particular domain. Coming on to my next slide, to understand health and medicine, we need to go at a molecular level. And when we dwell upon a molecular level, there are, four different biomolecules. These 4 biomolecules will define life. Example, if I want to define life, something which is living will execute 3 major characters. 1, it is growth. 2, it is survival. And 3rd, one's reproduction. So any organism which can execute growth, survival, and reproduction is been considered as living. However, to support this particular system of life, there are 4 major biomolecules. And these biomolecules are carbohydrates, proteins, lipids, and nucleic acids. Now, with these biomolecules, today's talk would streamline onto the central dogma. Now, when I talk about central dogma, this is a pathway which will define life. So the entire story of central dogma of molecular biology starts with the first molecule which we have learned from our primary school, that is the DNA. Now, DNA is your deoxyribonucleic acid. This is the molecule. This is the precursor molecule wherein all your replication, your duplication, your parental molecule, the template is being hidden into this particular molecule, what we call it as DNA. Now, this DNA, after some point of time, it will translate, it will transcribe into RNA, and RNA is ribonucleic acid, and from ribonucleic acid, it will get translated into a end product, which is protein. So I want to drag your attention wherein you have DNA undergoing replication, okay, and one DNA produces another DNA molecule. This is called as duplication or replication. Now, this DNA can undergo transcription, wherein from a DNA, it will get converted into RNA, which is a code. You can just imagine this RNA as a switch, okay, which will regulate this particular DNA, which will help in gene on and gene off. I mean to say, there's an upregulation and downregulation of DNA, which has been controlled by a molecule, which is called as RNA. Okay, and this happens through a process which is called as transcription. Now, the so formed RNA will get converted into its end product, and this end product is nothing but your protein. So if I want to tell you the central dogma of molecular biology, it starts with DNA and ends with the protein, and this hypothesis is called as the central, or it is called as classical central dogma of molecular biology. I have added a word as classical central dogma of molecular biology. Now, why did I add this word as classical? For a simple reason that as we started understanding these 3 molecules at a much deeper level, what we have understood is, these are not the only, central dogma, but however, this, based on this particular platform, a newer central dogma was being built up. And this is called as the modern central dogma of molecular biology. So the central dogma is now being divided into classical central dogma and modern central dogma. Whatever we have looked into this particular current slide is the classical central dogma, wherein I have DNA to RNA to protein. However, when I move forward, now I know it is not just the central dogma. There is something which is called as modern central dogma of molecular biology. So if you pay attention on this particular slide, what we have discussed is there is DNA, and DNA gets transcribed to mRNA or just RNA, and this RNA will get translated into a protein. And now what we know is this protein, this step will not stop here. There's a series of enzymatic reactions which will go on, which will metabolize the protein which has been produced. And this can lead into a production of anabolite, which is a product which has been formed during anabolism, and or a catabolite, which is a product which has been formed during your catabolism. So anabolism and catabolism can give rise to a sum total reaction of metabolism, and this is called as metabolite. So this proteome or this protein will end up with a metabolite, and this metabolite finally is responsible for a phenotypic characteristic of a given organism. So this is one part of the story wherein we started with gene, that is DNA, from DNA to RNA, RNA to protein, and protein to a metabolite, and a metabolite to a phenotype. So this is one part of the study. However, in today's discussion, what we are trying to assess is, when we spoke that DNA as an initial molecule, which is a precursor molecule, and this DNA molecule is governed. It has been operated. I want your 100% attention at this point. This DNA molecule has been governed with non-coding segments, or these genomic molecules are being governed with or being operated with certain non-coding factors, which will not become the integral part of your DNA, or which this component or this factor will at the end will not come into the structure of your protein, but however it will play a predominant role in regulating this DNA, which is the initial molecule. And these factors which will govern the DNA, which will operate the DNA without becoming the integral part of the DNA or without becoming the coding part of the DNA, these non-coding fragments are called as epigenetic factors. Okay, these are called as epigenetic factors. Now, please remember, what are epigenetic factors? Epigenetic factors are the non-coding fragments which will not become the integral part of your DNA, but however will play a predominant role in operating in the operational component of your DNA, and these factors are called as epigenetic factors. And studying of these epigenetic factors is called as epigenetics. Okay, I repeat, studying of these particular epigenetic factors which will not become a part or the component of your genome, okay, but however they will regulate, they will act as a memory to that of your DNA. Okay, so this is the genomic memory, and this genomic memory has been regulated by certain factors, what we call it as epigenetic factors, without being an integral part of the DNA. Now, studying these integral factors or studying these epigenetic factors, this branch of biotechnology is called as epigenetics. However, now this has defined or this has led into a newer dogma of molecular biology. Initially, whatever we have learned in the previous slide is called as the classical central dogma. Now what we have learned because of the advent of technology, this particular slide is called as the modern central dogma of molecular biology, wherein I study epigenetic factors, and study of epigenetic factors is called as epigenetics. And these epigenetic factors will govern your DNA, and study of your DNA is called as genomics. And this DNA will get converted into RNA, and study of these regulatory factors are called as transcriptomics. And studying these mRNAs or these RNAs will get converted into protein, and study of this protein is called as proteomics. And these proteins will further undergo metabolism, and studying these metabolism is called the study of these metabolism is called as metabolomics. And then this finally leads into the entire physical composition of the organism, which we study either in terms of phenotype or at a genotype level. Okay, so this we have understood very well in terms of your epigenetic factors and how exactly there is a transition from classical central dogma to the modern central dogma of molecular biology. At this point of time, if there are certain clarifications or if there is some query, I'll be happy to answer. Otherwise, I will move on. The concepts are clear. You can always respond with yes or no. I cannot see your chat box, so it would be better if you can, say yes or no.
Yes, sir. Yes, sir.
Okay,
Yes, sir.
Great. I am being a typical teacher, and the characteristic or the salient characteristic of a typical teacher is I want my students to respond. Otherwise, I don't want to have a monologue. I always would like to have a dialogue with my students so that I know that whatever we are trying to learn percolates into the student. Okay, chalo. Now, with this central dogma or this understanding of modern central dogma of molecular biology, now I want to draw your attention from just the word epigenetics, to the depth of epigenetics, wherein this epigenetic, as I told you, epigenetic factors are the factors which will not become the integral or which will not produce the protein at the end, but however, this will regulate your DNA. And this regulation is being controlled by your epigenetic factors, and very importantly, this has been controlled by post-transcription and post-translation modifications. Okay, and a predominant post-transcription and post-translation modification can happen at two important levels. One, it can happen on the genome, that is on the DNA, or it can happen on the proteins which are actually been wound with your DNA. And these proteins are tetrameric proteins, which are an integral part of your DNA, which are responsible for the stability of your DNA. These are called as histones. So these epigenetic factors can either govern your post-translation modifications such as methylation, acetylation, phosphorylation, glycoxylation, etc., on the DNA, or it can work
Please left the
on,
meeting aside, tell you.
or I request that, please mute so that, there is some decorum with the presentation.
और फिर देखो सर्टिफिकेट
Okay,
कैसे
so
मिलेगा?
okay, and then you have the histones, and these histones can be also regulated by your epigenetic factors. And there are two important enzymes, what we call it as HDAX and HATS. Okay, HDAX is HDAC, HDAX, and then HATS are HATS. Okay, so these are histone acetylases and histone deacetylases, which are very important enzymes which could be regulated through your epigenetic regulation. So now we know that epigenetics could rule the DNA as an epigenetic memory at two different levels. One, it could be on the genome itself, or the second one is it could be at a protein level, that is on the histone protein. Now, with this as an understanding, this regulation of your epigenetics could lead on to a change in the transcriptional complex. Okay, this change in the transcriptional complex could lead either it can become a promoter, this particular signal can become a promoter, or this can become a inhibitor, okay, through which a particular gene could be activated, or the switches could be activated, or the transcriptional complex could be downregulated. So the regulation, the epigenetic regulation could be studied at two different levels, one either at a genome level or at a histone level. For today's talk, more emphasis would be led onto the genomic level of the epigenetic regulation. So with this, let us try to move on. Now, as you have understood that we are referring to molecules. From molecules, we are going on to sub-molecular level, and at this point of time, we are not just on a superficial level, we are not just talking at a sub-molecular level, we are diving deep with regard to your sub-molecular level. So when I dive deep into the sub-molecular level, I require a particular branch, I require a particular tool which can help me, which can aid me to dwell deeper. Okay, so for this, I take the aid of bioinformatics. Now, what exactly is bioinformatics? So because the data, until now, this data was very simple. But however, now the data is becoming very vast. It is larger. It is becoming big. And hence I call it as big data. So since the data is humongous, and this cannot be manually controlled, or this cannot be manually studied. And hence, I require an aid of a tool which will help me in understanding this sub-molecular level interactions. And to help that, I would seek the refuge of the technology, wherein the best aided technology is your computer, which will help me in understanding my sub level molecules, and this will enable me for a commencement of a new era of biology. And this new era of biology, wherein I am trying to understand biology not just with wet experiments or wet lab experiments. I am also trying to understand the biology with dry lab experiments, and whatever experiments I perform on a dry lab, these are on a computer. These are called as in silico modeling, because the word in silico comes from silicon chips. The computer is made up of silicon and the microprocessors, and whatever biology I am trying to understand on these silicon chips that is on the computer, these models are called as in silico modeling. So right now I'm trying to understand epigenetics using computers, and this computers has led into a deeper understanding of biology. Now, using computers, if I'm trying to understand biology on a vague terms, this branch of biotechnology is called as bioinformatics. Now, what do I do in terms of bioinformatics? Bioinformatics is just a tool. Please remember, Okay, it is just a tool wherein it will aid me to understand, molecular interaction, submolecular interaction, sub-submolecular interaction, rather than me wasting time on my predictions, rather than me wasting time on my wet lab experiments. So this will actually help me in understanding whether my prediction or whether my hypothesis, whether this will lead into a null hypothesis or this can lead into an alternative hypothesis. And bioinformatics is one branch which will act as a kundali. Now, if two people are getting married, we look into their kundali, we look into their astrological component, but looking into an astrology or looking into the marriage kundali will not finally govern, the couple will be happy or the couple will be sad. But however, this will give me a vague idea. At least this will give me around 80 to 85% of an idea whether there is a compatibility between these two couples. Okay, so that's the reason why kundalis are looked into. In the same way, bioinformatics has been performed to look into whether there is a proper interaction of a ligand with that of its target, and what is the probability of my hypothesis wherein a given ligand can interact with that of a target. To understand this at a genomic level, I can use bioinformatics. At a transcriptional level, I can use bioinformatics. At a proteomic level or at a metabolomic level, I can use bioinformatics. And this means to say bioinformatics holds or it gets aligned with my modern central dogma of molecular biology, wherein I can build databases which are called as biological databases. I can use them for a comparative approach between organisms and between species and between phyla, class, orders, whatever you are trying to understand. And then this will enable me to develop tools, software packages, and operating systems wherein I can get my desired results. So depending on my dry lab data, I can start my wet lab data, and this will put up a strong foundation to understand whether my hypothesis or my thinking, whether this is right, whether this is feasible or this is workable. So bioinformatics is a tool which will help me in understanding my biology at a dry lab level. Okay, and then to prove it, I need to always switch on to my wet lab. That is my classical experimentation. Now, this is also to make you understand that when we talk about bioinformatics or epigenetics, do not segregate epigenetics from the rest of the composition, because it is not an indigenous system. It is not just one system. It is a multifaceted system, so that you need to think in 360-degree. You need to also have a moonshot view of whatever hypothesis you are trying to lay down and whatever hypothesis you are trying to prove it. So you cannot segregate this into a separate bowl and then look into it. You need to always have an integral composition, and with this integral composition of your analytics, of your molecular biology, of your wet lab component, and with your phenotype and genotype, you can look into the entire interactome and you can analyze everything using your computers. And this is the power of bioinformatics, which will come onto your table. Now, with this understanding, because we have understood bioinformatics, we have amalgamated this with biotechnology. There are 4 major revolutions which happened with regard to your bioinformatics. Okay, these 4 revolutions are called as blue biotechnology, red biotechnology, white biotechnology, and green biotechnology. So as the picture depicts, blue biotechnology is the marine biotechnology, what we are trying to understand using bioinformatics. Red biotechnology is the health and medicine, in terms of vaccine, in terms of disease, host, pathogen interactions, what we are looking into it. And then green biotechnology is the agriculture incorporated biotechnology, and white biotechnology is the industrial revolution, wherein you can use microorganisms to produce a molecule of a human interest. Example, insulin, wherein you can use recombinant E. coli to produce human insulin, what we call it as humulin. Now, with this 4 revolutions, today's talk will mainly dwell upon this red biotechnology, wherein we are trying to understand biotechnology, we are trying to understand bioinformatics, the application of epigenetics, and then how best we can understand this red biotechnology, or how well we can understand this epigenetics, which can actually help in minimizing suffering wherein we can develop certain diagnostic markers, we can develop certain therapeutic targets, wherein we can develop newer drug system so that, people who are suffering with various kinds of diseases and disorders, this suffering could be minimized. And hence, today's major domain of biotechnology would be red biotechnology. Now, what are the various applications of bioinformatics? I would drag upon a very important application, which is called as personalized medicine and molecular medicine. Now, this will enable me to customize. See, whenever I have a textile, whenever I have a piece of cloth, okay, so this piece of cloth cannot fit in everybody. So hence, what I normally do is there is a high possibility wherein I customize it according to my body fit. So the same way, you have plenty of biotechnology tools, and I actually customize it as per my requirement. And hence, you already have various kinds of medicine, but these are very, very generic. Now, usage of application of biotechnology or usage of epigenetic factors will enable you to customize any kind of drugs or drug targets, which will help you in understanding better molecular interactions in terms of customized medicine or tailor-made medicine, or in terms of molecular medicine. Now, with all these things, this has given rise, this understanding of our epigenetics has given rise to a beautiful subject. And this beautiful subdomain of bioinformatics is called as structural genomics. Now, in structural genomics or structural epigenomics, so this will enable me to understand and build, functional studies with regard to drug development and metabolic pathways. This also helps me in understanding one single cell and the interaction of this one single cell with various other microbiome, that is the sum total microorganism in my body, and how is this one cell interacting to the sum total, microbiome in my body. This also helps me in understanding the structure functional characterization, and this has led into the new family members of proteins. Okay, and I'm very happy that our lab, my PhD students are deeply involved, and around 7 proteins from Drosophila and humans. Okay, we have given up newer structures in terms of structural genomics and structural epigenetics. We have actually depicted these new structures, and these structures, all the 7 structures, have been patented, and these are now available at Swiss Institute of Bioinformatics, University of Basel. Now, this also helps me in understanding the biochemical structures in terms of crystallography and then NMR. Then finally, this will help me in develop beautiful targets. Okay, this will help me in synthesizing beautiful targets wherein the ligand interacts absolutely with that particular target, and this has led into better cure, better diagnostics, and better detection of any kinds of diseases. So with this, a newer branch of structural genomics has come up, and which is the major core area of today's talk, which is comparative epigenomics. So here, this is not just studying the genes which are present in humans, but this will also enable me to study the inter and trans species regulation at an epigenetic level. Example, now I can see how much of similarity is there at an epigenetic level between a human being and a chimpanzee, or a human being or with a macaca, that is a monkey, or the human being with that of a fish, a human being with that of another human being of a different geological point, or other human being at a different topological point. So I have various options. Example, I can find what is the similarity and dissimilarity, how many genes or how many epigenetic factors in terms of habit or habitat are similar between Indians and to that of Africans, or between Indians to that of Europeans, or between Indians and with that of Americans. So we are all humans, but on the way there is a huge difference in their microbiome, in their habit and habitat, and this can play a primordial role at an epigenetic level. As I have defined that epigenomics is nothing but a memory to a DNA. And maybe how do I make you understand this in a much better way? Let me try to take up an example that there is a person who has habituated for smoking. Okay, and for last 1 or 2 years he was smoker, and after some point of time he has quit the smoking, and it's been past 10 years. Okay, the person has quit smoking and at no point of time he is smoking. But however, since he has smoked for the last 2 years, like last maybe 12 years, so under that reason and under that condition, there are certain epigenetic factors which will remember that he was smoking 12 years ago, and after in the past 10 years he has not smoked. So there is a epigenetic memory which has been developed because of the smoking. So what I'm trying to make you understand is, for everything there's a memory which has been created at an epigenetic level, and that can be slowly transcribed into your genome. So a person who was smoking, after 10 years of quitting smoking, could develop something which is called as COPD. That is, you have a severe respiratory disorder, which is worse than asthma. These kind of people can develop COPD, and they might always say that I quit smoking 10 years back, but on the way I'm still suffering with a respiratory disorder which is called as COPD. So this can be because of your epigenetic memory. In the same way, when there is a replication of DNA, when there is a zygote formation or during fertilization, everybody gets a gift from their parents. Example, like you have a car, you have a bike, you have a land, you have a house, etc., etc. Likewise, there are certain epigenetic factors which are also gifted from your parents, either in a good way or in a bad way. If it is in a good way, then even your anxiety, your emotional quotient, your intelligence, all this could be a boon from your parents. Or on a negative side, if your great grandmother was having or suffered cancer, okay, so then your parents could not be, they did not suffer, they did not show any kind of phenotypic characters with that of cancer, but because of that epigenetic memory, those cancer traits could be expressed in your body, and hence the person, the third generation of F3 generation or F4 generation can still suffer with cancer, though they had very clean lifestyle, or though they had very clean habit and habitites. Okay, so there is the gist of this particular slide is to make you understand that there is something which is called as epigenetic memory, and we have to we have to budge for this particular epigenetic memory, and hence your entire phylogeny plays a very important role, not just your own personal phenotype. Now, with this all said and done, we have understood the importance of bioinformatics. We have understood the importance of why should I study epigenetics. So with all this background, I want to draw your attention in the work what we are doing from past 20 years. This is on neurodegenerative disorders. Now, to understand something, when I call it as disorder, we need to also understand, you have something which is called as a normal cell. Okay, from this normal cell, if there is some irregular impulses onto this normal cell, this irregular impulse will create disorder. They create symptoms, and from symptoms they will create disorder, and if these disorders are not being treated, they will get converted into diseases, and if these diseases are not being treated, they will get converted into multifaceted diseases, and if these multifaceted diseases are not being treated well, then ultimately this will lead into death. So this is the entire pipeline of how exactly from symptoms to death the entire pipeline operates. Now, our lab has been mainly dedicated towards neurodegenerative disorders, and we study especially 3 major neurodegenerative disorders, that is Alzheimer's, Parkinson's, and multiple sclerosis. These are the 3 major neurodegenerative disorders wherein we dwell upon, and trust me, with all 20 years of effort, we know nothing about them. Okay, we have published more than 150 papers, but on the way, we have patents, we have everything, but on the way, the disease is so complex. The disease is having multiple phases that we still don't know what exactly is the reason for this particular disease. Forget about the therapy, forget about a drug, forget about a vaccine for neurodegenerative disorders. We are at a stage wherein we still don't know what exactly is the causative factor for these particular neurodegenerative disorders, because some claim it is viral, some claim it is autoimmune, some claim that is habit is responsible for it, some claim that is genetic factors which are responsible for it, some claim it is habitat which is responsible for it, some people they claim that it is some researchers they claim that there are certain genetic disorders, there are some people, they claim that is epigenetic disorders, etc., etc. But trust me, my dear friends, right now, if you ask me what is the single cause for this particular any kind of neurodegenerative disorder, my answer is, I simply don't know. With this, a larger avenue, a larger door of opportunity has been opened up, not for designing a drug, okay, at least to design newer therapeutic, newer diagnostic, methods. I am right now, I'm not looking into what exactly would be the drug for Alzheimer's, what would be the vaccine for Parkinson's, etc. What I'm looking upon is, if somebody is suffering with, these particular diseases or disorder, can I have an early detection? Can I have an early diagnostic method wherein I know that, oh, this person right now is trying to develop certain conditions of, Alzheimer's or conditions of Parkinson's. So, this is the best method wherein either if it is not, preventive, how do I have an arbitrative effect, or how do I have an, curative effect? Okay. So rather than curative, right now we are looking into an arbitrative effect wherein I know I'm trying to understand what exactly is a, or what exactly is either a cause, or, what exactly is a diagnostic biomarker I can use it for Alzheimer's. Now, just to give a very brief upon, certain characters of Alzheimer's, there is a very important protein which is called as beta amyloid protein, and this beta amyloid protein will undergo changes, okay, at various levels. When this undergoes changes, at a particular area, it forms a plaque which is called as amyloid plaque, and once this plaque has been formed at a given area, okay, that area, diminishes with that, that particular activity. Example, if there is a plaque in my brain onto a visual area, so this means to say, whenever I see my daughter, whenever I see my parent, or whenever I see my, wife, I will not be in a position to identify them. Okay, no way I can identify them. But however, this plaque is only formed in my visual area and not on my hearing area. So when they speak out, I know that, it is my daughter or my wife or my parent. Okay, I know that, or my student, I know them, I can recognize them with with their voice, but not with my vision, because my vision, memory has been hampered. So this actually happens in your Alzheimer's. However, in Parkinson's disease, you have various kinds of tremors. Okay, there are various kinds of Parkinson's associated tremors, and there is an irregular impulse. Just imagine that you have put your finger into an electrical socket, and when you put your finger in your electrical socket, there is an impulse of current which comes into your body, and this can lead into an irregular mobility or irregular reflex, and this is one of the primordial factor in Parkinson's, and we don't know what exactly is the reason for it. But the third disease, and which is more dangerous disease, which is called as multiple sclerosis. Again, my dear friend, I really don't know what exactly is the reason for this. But however, what we have learned is, this people or the patients who are suffering, I didn't want to show images, I didn't want to show videos, because many times for an young budding population, this would be very, very disturbing. Especially multiple sclerosis, these kind of patients who are suffering with MS, the problem is they don't die, and they suffer for more than 20 years or 15 to 20 years. Now, under this condition, they become bedridden. There is a loss of their activity. There is paralysis on each organ. Okay, and when this happens, the patient goes into coma, and even under coma condition, the patient can survive for 5 to 8 years. So, there's a huge, emotional burden on the family. There's a huge financial burden on the family, and hence, all this put together can lead into a huge kind of trauma and suffering. And our kind of research, we are trying to minimize this kind of suffering, especially with the neurodegenerative disorders. We have established various kinds of models which could be used for bioinformatics and studying these kind of diseases, which starts with microbes, wherein we have various kinds of microbes. We also do something which is called as notobiotics, wherein we wash out the entire gut microorganisms, and we only inoculate only one microorganism to see what exactly is the unidirectional effect of that particular microbe. Okay, so we study a lot of microbiome with regard to neurodegenerative disorders, and then for looking into various kind of toxicity experiments, we use C. elegans as a model, which is a microscopic transparent nematode model, and a very favorite of mine is the fruit fly. just to tell you, around 8 Nobel Prizes, the highest Nobel Prizes are being awarded with the work with regard to your, Drosophila. So this is a beautiful human biomimic. Then followed by that, we have the mouse, we have the rat, and thanks to the technology of recombinant DNA technology and CRISPR, that we are able to develop various kinds of transgenics. Okay, so we do a lot of in vivo, in vitro, and semi-in vivo methods, wherein we are also trying to isolate, certain semi-in vivo methods, wherein we use RBC membranes, we use buccal cells, we use egg membrane model, and then try to mimic human diseases on these particular models. And then not to forget, as the classical mode, we always try to mimic under a control experiment, and these control experiments are nothing but your in vitro experiments, wherein we design the entire human system either on a microfluidic chip, or we design it using the polymer and the glass, and with this we are trying to mimic what exactly happens with human systems, and hence these are the various available models for human biomimics. So you can see these two are the renal stimulators, wherein for kidney diseases, we have tried developing synthetic urine and synthetic urine stimulators, and these will act as your two kidneys. And then we can also stimulate it. This is actually a dialysis machine which is being utilized for your in vitro systems. Okay, so what I'm trying to make you understand is, for studying epigenetics, you have multiple models. You have a huge buffet of models which could be set up and which would aid. It is not necessary that you need to directly take human tissue and then study, because there are a lot of ethical issues with the human body, and with that I can always go for a preclinical model or an in vitro model. So these are the various available models for me to study epigenetics. Done. So all these, whatever I explained till now, just to have a quick recap, we studied what exactly is biotechnology. We also understood, a beautiful branch of biotechnology, which is called as bioinformatics. And in bioinformatics, we wanted to understand a sub level of molecular biology. And hence, we did not want to understand just DNA, but we wanted to understand what are the non-coding fragments which are governing the DNA. And hence, we called it as epigenetics. And in epigenetics, there are various diseases and disorders wherein epigenetically it has been regulated, and our major focus for today was neurodegenerative disorders. Again, in neurodegenerative disorders, we studied there are 3 different neurodegenerative disorders, wherein it could be either Alzheimer's because of beta amyloid protein, or it could be Parkinson's, which could you have a tremor, a tau protein malfunctioning, which could lead into various kinds of tremor, and myelin or because of demyelination. Then we also studied something which is called as multiple sclerosis, which is again a multifactorial disease which can lead into slow paralysis of your vital organs. So these were the neurodegenerative disorders. And right now, what I am trying to make you understand is, how do I study these particular diseases at an epigenetic level? Now, today, because I know that most of the students are involved and most of the research scholars would be the audience for today. So what I thought is, let us try to understand a better epigenetic regulation using publicly available free databases. So publicly available bioinformatic tools, which are absolutely free, and they are available for you to into 24/7. Okay, any point of time they are available. So I want to drag your attention onto certain special kind of epigenetic factors. These epigenetic factors are called as microRNAs. Okay, so we're trying to understand various kinds of epigenetic factors. As I told you, epigenetic factors could lead into your post-translation modification of DNA methylation, phosphorylation, acetylation, glycoxylation, etc., etc. We also discussed something which is histone dependent, which is HDAC and HATs. That is histone deacetylases and histone acetylases. Now, apart from that, there's a special kind of a small non-coding, molecule, which is around 16 to 22 mer length. That is 16 to 22 nucleotide length. And these are the non-coding fragments. And these non-coding fragments, which can regulate your DNA at an epigenetic level, these are called as microRNAs. In short, they are called as miRNA. So today we are trying to understand miRNA, which is an epigenetic factor, one of the epigenetic factor, and how do I use various kinds of databases to understand these miRNAs. Now, moving on forward. So, from this point onwards, our entire intention of learning would be the epigenetic regulation in terms of miRNAs. Okay, how these non-coding fragments, which is around 16 to 23 mer or 25 mer, how exactly these can regulate your epigenetics, especially at a in association with your neurodegenerative disorders. So from this point onwards, we are trying to understand a collection of data which is specified onto miRNA, and these databases are called as miRNA databases or microRNA databases. To start with microRNA databases, the first database I want students to actually note down. If possible, I will try to share the link with all these particular databases at the end. So MIR base. So MIR base is another base. It's a database which is freely available. And in most of the conditions, I have made it a point that normally in our research, we use Schrodinger, we use Molegro, we use Discovery Studio, we use Discovery Environment to understand all these things. But, I wanted our BSc student, MSc students, and PhD students to experiment their data at a microRNA level or in an epigenetic level. So I'm considering all freely available databases, and one such freely available database is your MIR database. Now, in this MIR database, this is how the entire home page or the landing page of MIR database looks similar. And if possible, if you can take snapshots or screenshots, that would be easy for your later follow up. Now, this miRNA, that is MIR base, looks like this, and this is mainly used for miRNA sequences and annotations. And as you are able to see that there is the input which is required, and every microRNA, like everybody of us, we have a independent or a unique name. In the same way, microRNAs, these non-coding fragments, also have this independent names or unique names, and these are called as MIR base ID. Okay, so this, and here in the search, okay, or in the home, if you want to search a particular microRNA, then it is HSA. HSA is your human, okay, and then you need to specify in which organism you are looking at. Right now, in this particular example, HSA is human, and then MIR is you're looking into microRNA, and then you need to put up the name. So right now it is MIR, HSA, MIR 15A3P is the name of the microRNA what I'm trying to analyze, which I think that this is one of the microRNA which could be involved in Parkinson's disease. Okay, so I need to put the name of the microRNA. In the next slide, I'm trying to show when I input this microRNA 15A3P into the data, I click on that particular name, and then I can have options of which are the associated microRNAs with this particular microRNA, other microRNAs which are associated with 15A3P microRNA. So once I am done with this, you have the data, and it says that there is one result with the kind of input what you have given. And then when you look into the MIR-based data, it will tell you this is a sequence of the mature miRNA, okay, which has an accession number, and it will also tell you on what evidence is this particular sequence which has been curated. Okay, there are 5 experimentally cloned data. It's a clone data wherein there's an experimental evidence for this, and then it will also say that rather than just looking into one database, there are other databases which are also available, okay, wherein you can mix and match, or you can look into a better curation of your HSA, MIR 15A3P, and the other linking database is RNA Central. Then you have Tissue Atlas, then you have MIR Path DB. Right now it is version 2.0. Then you have the target miner, you have MIR RDB, and then you have the target scan. So why am I trying to make you understand this is from one particular MIR database to have a cross reference, you can always hop, you can always jump from one database to another base so that you can have a cross referencing of the same. Now with this, I know what exactly is the sequence, okay, and where exactly this particular sequence, okay, is being allocated in terms of its mature RNA, that is your MIR database. So now I know that this is a sequence and this is the accession number and this is involved with my Parkinson's. Now, but however, I want to understand much deeper, that is miRNA and which is the gene, because as I told you, the epigenetic factor will control your DNA. So DNA is nothing but a fragment of your gene. So I also want to know this miRNA, whatever I took up an example, I understand the miRNA, but I also want to know what is the association of this miRNA with a gene. So for this understanding gene target, I am hopping onto the second database, which is called as MIR-TAR base. Okay, I seek your attention. This particular new database is called as the MIR-TAR base. This MIR-TAR base, I have another database which is called as MIR-ROR base. Okay, so both they do the same function. Okay, wherein they will try to associate the so selected miRNA. They will try to associate that with a known gene. So your gene would be a target. Okay, your miRNA at this point of time would be a ligand. Okay, just to have a comparison. So you now know that this miRNA is binding to what kind of a target. So you will be able to understand well that how exactly the experimental validation with regard to miRNA and the target interaction is being related, and the entire data is actually being associated. So at this point of time, I want you to recall that in previous slide, I explained that you can always look into trans, inter or intra relationship species. Okay, within the species or outside the species, you can see how exactly there is a correlation. Example, I can see at what point of time on my skin or my hand, okay, long hairs got shortened into smaller hairs, and from fur to cilia. Right now, whatever hairs you will find on your hand, these are called as cilia. So initially it was very, very cold. Okay, and Antarctic, it was so much of lower temperature, so that there was a longer fur, there was a longer hair. So now I want to establish the epigenetic factors to understand when, at what point of evolution, the longer hairs, such as a bear or a polar bear, okay, from that point to a human, you have Australopithecus, Ramapithecus, Bryopithecus, Cryopithecus, and then Homo sapiens. So how did this entire evolution happen in terms of microRNA? Then I will be able to relate, and what is the gene which was responsible. So I'll be able to relate and validate the input, microRNA with that of the gene. So this is how from MIR-TAR base I will be able to export a gene and can look into that particular association. So this is how it will tell that this MIR-TAR, numbers or MIR accession IDs are now being connected to various kinds of targets. So now you know that, oh, this miRNA, if it has been present, then there is a correlation with cancer. Now, again, cancer is a big ocean. So to be very precise, there's a correlation with, cervical cancer, or if there's a correlation with prostate cancer, I'll be able to correlate the miRNA with that of the gene. Now, to understand, now I now know what is the miRNA. I also now know which is the target, but I also want to understand what exactly is the interactions. Okay, interaction of this particular miRNA with the target, and for that there's a beautiful database, and this database is called as TAR base. Okay, TAR refers to target base, which helps in understanding with the experimental support. It will help in understanding your miRNA targets on protein coding transcripts. So that is your transcriptional factors which will get converted into a protein. Those particular factors you will be able to understand. The only thing what you need to do is you need to go and look into a tab which is called as interaction, and then you can visualize. Then you can look into the statistics, and you can have various kinds of data mining tools over there. Okay, so TAR base will establish the link between miRNA targets with that of the protein coding transcripts. So what do I do? So this is the home page and various options. So here I need to select which species are you looking at. Do you want to have an experimental evidence for it, or it is just a methodology or a type? And also you can select what kind of tissue are you looking at. Example, if I am looking at prostate cancer, I will look into prostate as a tissue. If I am looking at a breast cancer, I will look into breast as a tissue. So, or myeloma, then I can look into your liver as a tissue. So, I can select a tissue. I can give my input. If you remember, there was an accession number. Either you can give in the name of the miRNA ID, or you can give in the name of the accession. Both are operatable, and you can also give the selected from the previous database. You can also give which is the gene what you are trying to select to look into the interaction. And now this tarbase will enable you to understand the interaction of miRNA with that of the gene, and this is how it will give you an experimental setup. Okay, so this is one, these are the targets, and in this targets, these are the miRNA which are involved, and these are the various kinds of experiments. These are the publication data wherein you have the literary data available, and then you have the cell line. This is from the cell line data, and then you have something which is called as micro T score. This is a statistical value which will tell you that if the score value is 0, then it is absolutely matching to that particular interaction, and if the score value increases towards 1, then there is a slow dissimilarity between the gene and the target. That is, the interaction is lowering in terms of gene and the target. Now, if you can see, you have a value of a micro T score value of 0.47, which is nearer towards 0, but however, a value of 0.95 is nearer to 1. So I can say that the above two, this HSA microRNA 181A5P is more similar or having more interaction with the gene of my interest, that is FLG2. Okay, this is having more interaction with my gene than compared to the microRNA 335 5P. Okay, because of a higher T score value. So this is how we look into it, and then you can select which are the microRNAs. What you can see, there is a plethora of data here for the gene interaction, microRNA gene interaction. You have, in this case, we have around 3,274 interactions, which has been supported with 46 experiments, which are later supported with 24 publications. And you can also see that there are 23 tissues which are involved with this particular microRNA and the gene. And then you can see that there are around 26 cell line experiments which have been established in 9 different cell lines. So for a researcher or for an MSc student who is planning for a dissertation, okay, so you can always take up this data and then build your hypothesis, build your epigenetic hypothesis, so that at a wet lab level, at a mimic level, whether it is working or not. Before you do your experiment, wet lab experiments, you can actually prove it using your dry lab. The next very, very important and interesting database is your MIR-RDB database. Okay, MIR-RDB database is a database which actually helps in predicting your target, function, and especially it will help in annotating your interaction. So it will help you in telling what kind of interaction. So this is the home page of it, wherein you need to put up an input of, I need to select which organism, then I need to also say which is the microRNA I'm looking into, and then which is my target gene. Okay, this is the input kind, whatever I need to select. Okay, I want you to go and read these 2 publications, that is in nucleic acid research and then in genome biology. Fantastic papers to read upon, so that you have a deeper understanding of how exactly MIR-RDB works. So with this input, next, if you go on to it, then it will tell you the various kinds of gene symbols. These are the genes, and then it will also give you a short description of what kind of the interaction is with the given microRNA with that of the gene. So once you understand it, then you can click on it, and then you can select. Example, I want to have all the interaction with tyrosine kinase 2. Now, if you are having an interest with oncology, then you know that in cancer, tyrosine kinase 2 plays a predominant role in your MAP kinase pathway and MEK pathway, that is mitogen associated protein kinase pathway. Your TYK2 plays a predominant role, and now your interest is to understand. See, you have around 7,297 predicted targets, wherein around 27 microRNAs are actually targeting your TYK2, that is tyrosine kinase, and with that entire plethora of genes, I can only stream down to 27 microRNAs. Now I know, okay, these are the microRNAs which are absolutely working on your tyrosine kinase 2. Okay, so as we also have understood the transcription, that is transcriptomics, wherein now I know that particular gene which has been controlled by a miRNA can also control your transcriptional factors. So as I told you, not all miRNA which have been bound or which have been targeting to a gene will be transcribed. Okay, so hence I need to also look upon whether what exactly is this interaction doing on the switch, whether it is regulating or it is inhibiting, whether it is acting as a repressor or whether it is acting as a promoter. To understand this particular gene transcriptional factor interaction, I am actually introducing a newer database, and this database is called as Trust database. TRR, pay attention on the spelling, TRRUST, and TRRUST currently you have the version 2, which TRRUST stands for Transcriptional Regulatory Relationship Unraveling by the Sentence-Based Text Mining. So, this will actually take up the aid of your literature, and then it will try to, screen for your transcriptional factors. Very, very useful database. Right now, I am looking into CDKs, that is cyclin dependent kinase 1A, as an input. Okay, and then I am seeing what exactly is the gene interaction. And when I say this is my input, and then when I look upon this, then it gives me a data which says CDK, cyclin dependent kinase. This is actually interacting with so many different proteins. So this will also give you an idea of not just the gene miRNA transcriptional regulation, it will also give you an idea of what exactly is miRNA gene transcriptional protein interaction. So slowly you're trying to understand the entire modern dogma of molecular biology. So once you have this, okay, the next part is to understand, rather than the gene and the transcriptional level, I also want to know how exactly miRNA and transcriptional level, interactions have been, going on. And for this, what we do is there's another beautiful database which is called a TransMirror database. And this TransMirror database will help in understanding networks, functional networks, annotation networks, have been, or curative networks. Okay, this has been much more helpful in understanding, the interactions. And what you need to do to develop networks is, here you have an option called as network. You need to select network. You can also go for enrichment and prediction. But however, to develop a newer network, you can go on to, hello. Okay, another 10 minutes, I think we are done. So you have the TransMirror database, and with TransMirror database, you can also look into the miRNA and the transcriptional factor interaction. So what you need to do is, you need to just select organism. Okay, you can go for rat, you can go for C. elegans, then Arabidopsis thaliana, you can go for Drosophila melanogaster. Okay, so then select them. You have two options. One window you need to select the microRNA, the other window you need to select the organism, and when you do it, you get a beautiful network. It's a complicated network. You need to actually sit down and then analyze the network, which will take more than, depending upon the number of interactions, that will take more than 10 to 15 days. For us to understand one network, established network, we take more than 1 month to understand this particular network, because the data is huge. Okay, and everything is interacting, everything is, one is activating, one is regulating, one is will act as a repression. So hence, understanding these network at a holistic level becomes very, very important. So the next point is, okay, I have a gene and then I have a transcriptional factor. This is getting bound. There's an interaction which is well established. All this is fine. But is this, because of this interaction, is the miRNA getting expressed? Is the miRNA getting upregulated or downregulated? To understand this on a particular target, there's a beautiful database which is called as MITED database. And this MITED database, you can put up multiple microRNAs together and then read it at one shot. So you can go for, multi-query, or you can take top 10, top 5, top 3 microRNAs and also analyze, which will help in understanding. Please remember, it can help in understanding what exactly is the expression of the microRNA in a given tissue. because we took up an example of prostate cancer, or we took up an example of Parkinson's in your in your brain, so or your brain tissue or your hippocampus. I can study whether that particular microRNA, whether it is having a regulation or not. This part I can actually understand using this MI10. So this is how the MI10 database looks like. You need to have your input entries. Okay, that is, this is the name of your, or the accession number of your microRNA. Then you need to select which kind of a tissue. It could be brain, adipose, or whatever tissue you're liking to. And then you can also correlate to that of a disease. You can actually select what kind of a disease you're looking into. And then when you press enter, in the next point, it will tell you the expression patterns. You can see that here there's a lower expression. Here you see there's a larger expression in the blood, and you have a higher expression in the brain. So the brain expression is somewhere around 6, and the blood expression is somewhere around 5. And then it will give you the entire details of what exactly is the Sankey plot of the tissue and the disease correlation using this particular database. So with this, I have introduced you to multiple, databases which can study just miRNA, which can study miRNA with a gene, which can also study the miRNA with the transcriptional factor, which can also help you in understanding the gene with that of the transcriptional factor. And finally, you can also associate your miRNA, transcriptional factor, your epigenetic factor with that of your disease and tissue type. So with all this, you can build up your story. Okay, it could be whatever story. Okay, you can always correlate to the epigenetic regulation. With this, I need to show you that this particular whatever databases we actually learned today, this has very recently we published 2 papers. This both the papers are in the year 2024. Now one is International Immunopharmacology and the other one is Neuroscience Letters, wherein the Parkinson's disease, whatever I was talking about, you can just read the title, which talks about the miRNA, transcriptional factors, and high-risk genes, okay, which are involved in the regulatory landscapes of your Parkinson's disease. Okay, and then again there is a computational network pharmacology approach, especially with Parkinson's. These are the 2 papers very recently we published. So what am I trying to make you understand with these papers? What I'm trying to make you understand is using a beautiful hypothesis, and then with the freely available databases, just with building a very, very strong story, okay, you will be able to publish. So I seek, okay, students from various disciplines, wherein you can publish well, okay, with these kind of freely available databases. Not necessary that you need to have always Schrodinger or Molegro or Discovery Studios. These are very easy to understand. The only thing is you should be extraordinarily strong with your biology. Only when you're extraordinarily strong with biology, the tool bioinformatics could be used to understand the same biology at a sub-molecular level, which is nothing but your epigenetic level. We have studied with Parkinson's. You can study with any kind of disease or a disorder, build a story, and very importantly, I appeal to you that you need to publish. In science, if you don't publish, whatever your science you do, it's of absolutely no use. So hence, whenever you are publishing, publish it at a very good, with a very good publisher. This will add on value to you. If you are planning for a MS, if you are planning to, your higher studies, your PhD and other things, fellowships and other things are being attracted with publication. This is an evidence to show that, the candidate knows things, at a better level. Okay, so there is the credibility of the candidate actually increases with these kind of publications. I thank Pratik Jain. Mr. Pratik Jain is currently I have 4 PhD students, and Pratik Jain is like super interested with epigenetics of a disease, of a skin disease which is called as atopic dermatitis. He is in fact working on atopic dermatitis, especially in terms of developing AI tools, using ML tools, and using various kinds of epigenetic regulatory markers for understanding skin disorders, that is psoriasis and atopic dermatitis. So this brings to the end of my talk, wherein I work as an adjunct professor and a chief scientific officer at a well-established school which is called as the Omics School, which is in short it is called as TOS. wherein most of the training programs, consultancy, dissertation, hypothesis, synopsis planning, and most of the the grant writing and publication writing, okay, we take up as a consultancy services. So if any point of time, if you require any kind of an help, so I work as a adjunct professor and the chief scientific officer at TOS. So the email is theomicschool@gmail.com, and these are the contact numbers. You can WhatsApp, or you can just leave a message, or drop an email. So we will contact you and we'll see how best we can help you in terms of scientific aid. So with this, I thank the entire audience, okay, who are together and who are patient enough in understanding these particular databases, and then to correlate to various kinds of diseases and disorders. I thank the entire college and especially RUSA for funding the entire webinar, and special thanks to Ruchika madam and the principal of the college, who has facilitated the entire talk. Thank you very much. That's my time. Thank you very much. Over to you, sir.
Thank you so much for your knowledgeable information, sir. I would request the participants to post questions if they have any questions related to the topic. They can post their questions in the chat box, or they can open their mic and directly ask. Okay, so
the better option is you ask, rather than messaging. Okay, I always I'm a lively person, so I would always like to talk to people rather than talk to their messages.
So there's one question, sir.
Yes, sir.
From one student. That is, how do you see the future of bioinformatics evolving in the field of epigenetics and neurodegenerative disorders? Are there any emerging technologies or methods we should be aware of?
Obviously, sir, all these are like budding technologies, especially in India. Most of our curriculum is outdated, and with the autonomy which has been given, we need to upgrade our students. Especially, we are very conservative in terms of learning. Okay, and we need to be open for various kinds of learning, and hence I urge students for learning coding, amalgamating that with statistics, amalgamating that with computer science, mathematics. Okay, we can develop beautiful mathematics, especially physics and chemistry, all put together. Okay, you have a huge scope. So do not just think as biotechnologist. You need to bring all the subjects to one table. Okay, and then try to understand this in a multi-dimensional viewpoint. So there's a huge scope, extraordinary scope. But however, for that you need to have a very, very strong foundation with your biology, which obviously most of your biotechnology students will have. But upon that, you need to always have layering in terms of internships, in terms of dissertations, in terms of your higher degrees or certification, which will help you in understanding, or which will help you in understanding the better components of, bioinformatics, especially in terms of epigenetics. Just to tell you, there are no diagnostic markers for Parkinson's or neurodegenerative disorders. In this particular research, we have come out with certain novel miRNAs, which at an inception level, at a zygotic level, we can tell whether if this particular miRNA has been present, then the developing fetus could develop after some point of time, though it would be, at a window period for some point of time, it will later can develop into, a disease or any kind of neurodegenerative disorder. So your epigenetic components can act as a diagnostic tool for early detection of your diseases and disorders, be it any disease or, any disease or disorder. So that way, I see a tremendous amount of scope with epigenetics and bioinformatics, sir. Over to you, sir.
So there's one more question.
Please.
What role does network analysis play in understanding miRNA interactions and their impact on neurodegenerative disorders pathway?
Okay, good, great question. So normally, when you look into interaction, because as I told you, we cannot segregate them. We cannot segregate them into multiple, chambers, because everything is interconnected. If concepts, these, when I say miRNA 32P, this miRNA 32P does not have only one function. It has multiple functions. So to understand what exactly are the multiple functions of the same miRNA. Okay, so I need to understand what are the various kinds of interaction wherein one the same miRNA at one point of time in a given tissue can work as an activator. The same miRNA at the other tissue can act as a repressor. So to understand what exactly is the function of that miRNA at a given tissue connected to a particular disease or a disorder, I need to understand what exactly is their interactions and their associations. And because of this reason, okay, not just with neurodegenerative disorder or Parkinson's, take it any disease or disorder, I can use miRNA as one of the diagnostic target. And hence miRNA plays a very, very important role. So here, with the interest of time, we just explain in terms of miRNA. There are various other databases. There are various other epigenetic biomarkers also. Example, which can work on chromatin, which can work on the stability of the chromatin, which can work on your histone proteins, which can work on your post-transcriptional and post-translational modification. There are entire plethora of epigenetic factors. It is not just miRNA, but miRNA is one of the example wherein you are able to understand that epigenetic factors play beyond just being a non-coding fragment. It plays as a memory, okay, onto the DNA.
So there's one more question posted
Yes.
by Ariba. Which coding language should I start with in order to pursue bioinformatics in future?
Okay, great. If you are a biotechnologist and if your foundation is in biology, okay, your primary foundation is in biology, I would refer the easiest coding, because I don't want to start with something which is very highly complicated. So for me, if you ask, if I am a BSc or an MSc student, if I am interested with learning coding, so I would always prefer Python. So once you learn Python, from Python you can go on to BioPython. Okay, this will enable you, because you have certain advantages with Python because it has various kinds of suits, okay, which will actually help you. And then a very huge advantage is most of the codes are freely available. You have something which is called as Python community, and onto the Python community there are n number of people who are ready to help you, and coding becomes much more easier, especially for a beginner. So I would always refer Python to start with. However, you have other modes which is called as Perl, and then you have BioPerl, then you have something which is called as Java, then you have BioJava, okay, or Oracle, BioOracle. Okay, so for me, the easiest way to learn is Python. Okay, and then upgrade it to BioPython. That's sufficient.
Yes, Mr. Sandeep Nigam, you can unmute yourself and ask the question. Sanjana, you can also unmute yourself and ask the question.
Hello, good afternoon, sir.
Very
I
good
had
afternoon. How are you?
I'm fine. I wanted to ask a question that has the extent of epigenetics in progeny been quantified, or in any way can it be quantified? Like you took
Absolutely,
examples of.
absolutely, ma'am. Ma'am, in the first slide wherein we spoke about the sequence of that miRNA, I can take that miRNA in a given tissue, I can design a primer and do a simple PCR or an RT-PCR, wherein in the wet lab experiments I can prove it.
Yes. So, like, what I had thought, like, you gave the example of cancer and even smoking. For example, if there's a person who is a severe alcoholic by their own choice, and if
Correct.
this person goes ahead and has children, can that child just have a genetic predisposition to alcohol addiction? Like, is it that powerful enough to
Yes,
be
yes, it is
transferred?
absolutely powerful, ma'am. As you said, yes, we can actually look into it. You can actually look into it, ma'am. Okay. And if you want to dwell deeper, there is something which is called as MIDIP. Okay, wherein you can have a MIDIP is you go for a methylation sequencing pattern, or you have a bisulfite sequencing pattern. You can go onto the whole genome of those kind of organisms, those kind of fetus. You can just take one cell and analyze it. Okay, that's the power of miRNA. And you can see what exactly is the epigenetic variation, and that baby or that particular fetus or a developed baby, okay, what can happen to the epigenetic regulation after 20 years, after 30 years, after 50 years, this could be analyzed. And a great example, if you remember, Now, 4 to 5 years back, I know that most of you know Hollywood heroine called as Angelina Jolie.
Yes.
Angelina Jolie had, she was detected. And this particular branch of early detection of diseases is called as pharmacogenomics. Using pharmacogenomics, she was able to know that before the development of breast cancer, she had certain genes which have been transmitted from her mother, which in due time can be regulated towards breast cancer. So she went on to mammography, and under mammography, and then when she did a tissue biopsy, those cancer causing breast cancer causing microRNAs were upregulated, and that's the reason both the breasts were amputated before the onset of cancer. So this can be easy detection method, and right now in India, not many people are using these particular kits. We still rely on enzymatic kits or ELISA kits because the technology is a bit expensive. But however, if we can work out with the technology and the costing part, then this can actually change the entire revolution of diagnostics in India.
Okay.
Thank you for that question, ma'am. Very beautiful question. Thank you.
Thank you, sir.
So there's one more question.
Please, sir.
How can I obtain data on neurodegenerative disease, considering the difficulty in accessing patient data?
Okay. Again, there are two things. When it comes to preclinical data, that is experimental models. Okay, so you can actually retrieve data from the biological databases. But however, when you have the patient data, okay, especially with certain kind of neurodegenerative disorders, don't just consider neurodegenerative disorders because it is a big ocean. So I would always prefer to have a specific approach, a focused approach on one kind of a disease. Okay, so just take an example of Alzheimer's. So if I'm taking up Alzheimer's, again, you need to check on which kind of Alzheimer's can happen for various kinds of things. So you need to be extraordinarily specific of what exactly you're selecting, because bioinformatics works with one mantra. The mantra is garbage in is garbage out. Okay, so if I am asking a non-specific question, the computer will give me a non-specific answer. Okay, so I need to be extraordinarily specific with my mantra, wherein I use a specific query and the computer also will give me a specific answer. So with this, I should know what is my disease and specifically what kind of a disease is that. And then very importantly, for patient data, the best database would be from your NCBI. Okay, so you have something which is called as CLIN database. CLIN refers to clinical database, and in that clinical database, extracting data from a given population becomes easier. But again, you need to look upon what is the geographical point and the topological point of the patient, because you cannot take up American population and then compare it to Indian population. You cannot consider that as Indian population. So you need to, when you are retrieving data from the database, you need to define, okay, what is the nature of your patients or your data. Okay, if you are selecting, or if you want to compare the miRNA from the Americans, between Americans and Asians, then you can do it. But you cannot take one miRNA from Americans and other miRNA from Africans and other miRNA. So there is the data is not redundant. Okay, it should be, it should be able to match. So when you're selecting from the CLIN database, that is clinical database from NCBI, you need to take care of this particular data, and also you need to see whether you are taking MySeq data, HiSeq data, or it is the entire global genomics. Okay, all this has to be well defined before you pick it up. The best would be to select Illumina from an Illumina platform. The best would be to select the entire HiSeq data, especially for clinical diagnosis. Okay, I have we have a room for a last question, if you have any.
So how can one predict and determine unknown protein that might be causing various neurodegenerative disorders?
Great.
Using known epigenetic data.
Okay, so if you in between we spoke about how exactly a gene can interact, or an miRNA can interact with protein. Then we discussed with regard protein databases. So a better version to understand protein database would be your string database or stitch database, okay, which will enable you to see, if this particular gene is getting transcribed and then translated, you have the protein. So you can do something which is called as a reverse engineering. So you already have a protein, and with the protein, you can see which is the RNA which has been binding to that particular protein. And from the RNA, you can go back to your DNA. And from the DNA, you can go back to your epigenetic factors. So it is, rather than forward, you go on a reversal step, and you can trace which are the proteins which are getting affected because of your miRNA. This is called as miRNA protein interactions. This could be aram se done. For all this kind of approach, we use a paid software which is called which is from a company which is called as Qiagen. Qiagen, Qiagen. Okay, and they have developed a beautiful paid version of the software which is called as Ingenuity. So Ingenuity is a software which will help in understanding these kind of databases. But apart from that, you can have multiple different kinds of step-by-step databases which will not have one complete package, but according to your requirement, you need to use different kinds of databases. But still, on a publicly, freely available platform, you can analyze, reverse engineering or protein to miRNA interactions. Okay. I think with this, if you have multiple questions, you can leave a message at theomicschool@gmail.com. Okay, and or you can WhatsApp us on 90196226649, and I will be happy to answer them. Okay, with this, I thank everybody who has directly or indirectly contributed for this particular webinar. So I thank the host for that beautiful, humble introduction, and Ruchika Dalvi ma'am, who have actually corresponded in all the best ways. And in fact, from past 2 months, she is having an hold on me, and always at any point of time, she was like on check to know. She was my transcriptional factor, and she was actually deciding, what time, when, what is the title, etc., etc., etc. And thank you very much for the principal who has given this opportunity, and the RUSA coordinator who has facilitated this particular webinar. Thank you all my dear friends who have attended. I see that there are more than 100 people in the webinar. So, thank you very much, everybody. And hopefully together we can learn, better biology, so that in the society we can minimize suffering. Okay. So thank you, everybody. One or two people can unmute and, just give me your feedback. That how did you feel about this particular 1 hour, because your 1 hour was invested. So just any 1 or 2 people can actually, give me your feedback. Anybody. How did you feel about this particular webinar? Or did you understand? Were you able to follow? Any 1 or 2 people can.
It was very informative, sir. Thank you, sir. Thank you so much.
What is your name?
Vicky, sir. Vicky.
Vicky. Thank you, Vicky, sir. Thank you for your kind words, sir. Thank you, sir. Okay, and one humble request, Vicky, sir.
Sir, I am Dr. Bhuvneshwari. It was very wonderful because it was very new also. The topic was very new, although I have worked in miRNA, but miRNA based database is very new. So we want some interactive hands-on training, so that we will be knowing, and we will be knowing how to perform it, and then we can publish paper.
Absolutely, ma'am.
Thank you very much, sir.
Yes, ma'am.
Thank you very much,
ma'am. Thank you very much for your words, ma'am. And any point of time, you have my phone numbers. So any point of time, if you require any kind of an help, it's not necessary, it should be organized through a webinar. Any one person also, if you require any kind of an help, please contact us and we'll be able to help you out, ma'am.
Thank you very much, sir. Thank you very much.
For your student, it could be for a publication, but please publish.
Yeah, sir. So we can, very nice to, we will be contacting you, sir.
Yes, ma'am.
Thank you very much.
Thank you, Bhuvneshwari, ma'am. Continuing on to Vicky, sir. Vicky, sir, I would request you to actually log on to all these databases and then, explore. Just explore. Don't find any data, don't have any relevance. Just with a plain mind, try to explore it and see how best, your mouse will work. Okay, just click on data, and then you will have something new. Okay, and you will be happy for it.
Yeah, sir, definitely.
Thank you, Vicky, sir.
Thank you, participants. Feedback link has been posted in the chat box. Kindly give your valuable feedback, and I'll ask our next faculty member to give the vote of thanks.
I, Ms. Ayushi Mishra, on behalf of Patkar-Varde College and Department of Bioinformatics, feels heartful of gratitude and admiration for the remarkable journey we have undertaken together in this enlightening webinar. It is truly an honor to address such an esteemed gathering of individuals who are deeply committed to advancing our understanding. I would also like to express my profound appreciation to our distinguished speaker, Dr. FARHAN ZAMEER, sir, who generously shared his expertise, insights, and breakthrough findings. I begin my vote of thanks by addressing our appreciation to the management of Chikitsak Samuh, Chief Education Officer Dr. Mala Kharkar ma'am, our Principal Dr. Pratibha Gaikwad ma'am, Chief Coordinator of Sciences and Coordinator of Microbiology, Biotechnology and Bioinformatics, Mrs. Ruchita Dalvi ma'am, and to our sponsoring organization RUPSA. I extend my heartfelt thanks to each and every participant who joined us from across the country, bringing with them a wealth of knowledge, diverse perspective, and unwavering dedication. Behind every successful webinar is a team of dedicated individuals who work diligently behind the scenes. My gratitude goes out to the teachers who ensured the seamless execution of this event, allowing us to come together in a virtual space where ideas and knowledge flow freely. As we depart from this webinar, let us carry forth the torch of collaboration, innovation, and empathy that has illuminated our discussion.